Hi, everybody. This is Ms. Fennell back with you again today to talk to you about genetics. And genetics is the study of how heredity and how DNA is passed on from one generation to the next. The first scientist who is credited with doing genetics experiments is this guy who you have been doing a little activity with, the before and after activity, by the name of Gregor Mendel. For the longest time, humans realized that heredity existed not only in human beings, but also in livestock and plants, and offspring tend to resemble and have similar traits and characteristics to their parents. But Gregor Mendel is the first scientist credited with actually trying to figure out how all of that worked. He was from Europe in a country called Austria, and he did his research when he was a monk at a monastery, which is similar to kind of like a priest, if you kind of think about that. And he was very interested in science, and so he was the monastery's gardener. And as part of his gardening duties, he also got to conduct research. And so he published his data and all his experiments in 1866. He did a lot of work. I'll talk in just a moment about how he was cross-pollinating his plants to do his experiment. It was excruciatingly tedious work. It took hours and hours and hours. Sad story about him is that he publishes his work. He shares it with other scientists. And at the time he shares it, nobody understands what in the world he's talking about. So he goes back to living his life quietly at the monastery. He dies at age 61, never realizing that he is going to be part of science books that people will be studying, 100 and some years later. His work was kind of rediscovered and figured out by other scientists in the early 1900s, quite a few years after he passed away. So today we call him the father of modern genetics, and his experiments that were at the beginning of us understanding how genes were passed on. Heredity, as I said, is the passing on of traits from one generation to the next. Mendel had no idea what caused the heredity in living things. Of course, today we know that it's DNA, and it's the DNA that's getting passed on through reproductive cells that leads to the heredity of different living things. Traits are characteristics that can be inherited in the genes. So today and the rest of this week, we're going to be doing genetics problems with various traits. So it's any kind of a characteristic that is determined by the living thing's DNA or genetics. In the case of Mendel, he is famous for studying peas. That's right, folks, pea plants. This is how he unlocked the secrets of genetics. And he noticed as a gardener that the peas he was working with had lots of different traits, lots of different characteristics. Some of them have purple flowers. Some of them have white flowers. Some of them have their flower on the side. That's called axial positioning. Some of them have their flower on the end. That's called terminal. Some of them had yellow seeds or green seeds, or some of them were tall and short. In the case of pea plant height, it's not like human height where we have tall and short and everything in between. In the case of pea plants, they were either tall or short. There was no like medium or in between or that kind of thing. And so he actually did experiments on these 7 traits or characteristics of peas. And so, as I mentioned before, his work was tedious, and he worked with cross-pollinating peas. And if you remember, flowers are the reproductive structures of plants. So you have the stigma in the middle of the plant, that's the female part of the plant, and then you have the anther over here, which is part of the male part of the plant that produces the pollen. So if you take pollen from the plant, the pollen has to travel down the stigma and fertilize the eggs that are at the bottom here. And that's how seeds are produced. And so flowers their function is to make seeds, and plants do sexual reproduction. So what Mendel would do is he would remove the anthers of one plant so it could not self-pollinate. And then he would go over to another plant and use a paintbrush to get a little pollen on his paintbrush and then move the pollen to the plant that he wanted to cross with. So I'm going to be saying here that he's crossing plants and all of that. And this is how he did it. He took pollen from one plant and used it to fertilize the eggs of another plant. I'm pretty sure I can just picture Mendel out in the garden for hours on end with his paintbrush pollinating his plants here. And I just have to believe that people who lived with him might have thought he was crazy because, there's Mendel out with his paintbrush with his pea plants again. It's kind of a funny thing to do. I would love to talk to them and see what they thought about him. So I'm just going to tell you a little bit about Mendel's experiments and what he did and why that's so interesting. So what Mendel did is he first, oh, this distance learning stuff, you guys, I don't know. All right. So he first took what we call purebred tall plants. And he called them purebred tall because they came from parents who were tall and grandparents and great grandparents, if you will, plant wise, that were all tall. And he cross pollinated those and bred them with purebred short plants. And he wanted to know what would happen. So he took this plant, and let's just say that he's using the egg from this plant, and let's just say that he's using the pollen from this plant. So he's taking his little paintbrush, getting the pollen here and going over to this flower and pollinating it. When he did that, he pollinated it, he waited for seeds to form, he planted the seeds, waited for the plants to grow. And the interesting thing about that is he got 100% tall plants from that cross, when he crossed the purebred tall with the purebred short. Now, at this point, he doesn't call these purebred pea plants because they have a tall parent and a short parent. So we're going to call these hybrid plants. Hybrid means mixture. They have a mixture of a tall parent and a short parent. So then Mendel gets curious about it, and he wonders, what if I let these hybrids self-pollinate? All right. So then what if I take a hybrid tall and cross it with a hybrid tall plant? What do you think we will get? Some people would say, well, I think 100% tall. By the way, if I go back to this one, did anybody think you were going to get medium plants? I guess I already said there's only tall and short plants, but you cross a tall with a tall. He got kind of an interesting result. He got 75% of the plants to be tall, and he got 25% of the plants to be short. And so he starts wondering why this is. How is it possible to mix two talls and end up with short plants? Where is this short coming from? And his hypothesis was that these hybrid talls, this was a tall pea plant, but somehow it must be carrying the gene for shortness. And so he starts thinking about this and comes up with an explanation about why you could cross two hybrid talls and end up getting 75% tall and 25% short. By the way, he does this, just to go back for a moment here, he does this with all of the other traits he was studying. When he crossed purple flowers and white ones, he got 100% purple. But then when he self-pollinated the hybrid purples, he got 75% purple, 25% white. He ends up calling the trait that shows up when I cross a purple and a white, I get purple. He calls that the dominant trait. So we still call the trait that shows up, or the gene that shows up in a cross like this, the dominant trait. And we call the one that's hidden the recessive trait. That's what Mendel called them, and we still call them that today. So basically, we have figured out a lot more. Mendel had an explanation for this, which I'll share with you here in a moment. And we agree with him still, and have added to his knowledge as well. So a gene, remember from our last chapter, is a piece of DNA that gives directions for making one protein. It'll control a trait is called a gene. Remember from our last chapter, we go genes to proteins to traits. So the gene is directions for making a protein that results in a trait. For example, if you have brown eyes, you have a piece of DNA that gives directions on how to make a protein called melanin, and that melanin is what gives brown eyes their color, the trait of having your brown eyes. If you have blue eyes, you happen to have inherited a gene that does not make melanin, and that leads to the trait of having blue eyes. Eye color is actually inherited by it's caused by two different genes. And maybe at the end of the chapter, we can get more into that. Allele is that difficult word from the reading, and don't let it scare you too much. It's just different versions of the same gene. So in pea plants, we use the big T to represent the tall version of the gene. Plants that get this gene will be tall. And in pea plants, for this particular trait, happens to be a case of complete dominance, where the dominant gene will completely cover the recessive gene. We use a lowercase t to show that it's a different version for the same trait for the short allele. So we don't use an S, we use a lowercase t to show that the tall is dominant over the short. We also know, just a quick review, that living things have reproductive cells. We have eggs and sperm that are formed by meiosis. That's review from last week. They each have half the number of chromosomes so that when these two cells come together. So it's not even just a random number of chromosomes. You have in the human body, for example, 23 chromosomes in the egg and 23 chromosomes in the sperm. When they come together, you get a grand total of 46. And so if you remember our karyotyping way back from a couple of months ago, was it before Christmas? I don't know. But we get 2 copies of every chromosome. And it turns out in each pair, one of your number 1 chromosomes comes from the egg cell the person got from their mother, and one of their number 1 chromosomes comes from the sperm cell they got from their father. So when we say that the reproductive cells have half, it's not just a random half. Egg cells have one number 1 chromosome, one number 2 chromosome, one number 3 chromosome. Another trait of these chromosomes is they carry genes for the same trait. So I don't know for sure, but let's say the gene for blue eyes or brown eyes is on chromosome number 1. You're going to get one version of that gene on the chromosome you inherit from the egg cell from your mom, and you're going to get one version of that gene on the chromosome you get from your dad. And so because of that, the big thing you need to know here is living things, not just humans, but all living things that have 2-parent reproduction, they get 2 copies of each gene. They get one gene from their mom and one gene from their dad. And depending on what happens, if it's dominant or recessive mixtures that they're getting, that will determine their trait. So if they inherited a dominant gene from each parent, they'll be what we call purebred dominant, where they have the same genes and they're dominant. Or if they inherited 2 short genes, in the case of pea plants here, we call that purebred recessive. And if they inherited, a dominant and recessive from one parent for each one, we call that a hybrid. So there's really only 3 possibilities. You could have inherited 2 dominant genes, 2 recessive genes, or one of each from the parents for each trait. So the cool thing about this is we can actually use this to make predictions. We can do these, what I think are kind of fun little puzzles called Punnett squares. So if I go back, so I'm going to show you how to do Punnett squares here. This is the important part you really want to pay attention to. So if we go back to our little video here, and I showed you how that Mendel crossed a purebred tall plant with a purebred short plant, and he got 100% tall. I'm going to try to explain to you here how it is he was able to get that. All right. So the purebred tall plant, we are going to represent with the genes big T, big T. It's purebred tall. It has 2 copies of the tall gene. And Mendel was crossing that with what we now call a purebred short plant. So this is the purebred tall. And this is the purebred short. And again, he was using the egg cells from this plant and the pollen cells from this plant to cross them. So basically, each plant was getting a dominant T from one parent and a lowercase t, a recessive gene from the other parent. Because they had inherited the dominant gene, they will have the dominant trait. And that's how we got plants that were 100% tall. Okay, now it gets really complicated. I'm not going to confuse you, but when we start to have different letters, it can get real messy here. So a Punnett square is a way to basically show you this and what are the possibilities in the offspring if we cross 2 parents with different kinds of genes. All right, so in a Punnett square, what we do for this cross, and again, we use it to make predictions. We're going to start with a square. And in 7th grade science, we only do 4 square Punnett squares. You may go on where you do 8-celled ones or 16-celled ones. It gets a lot more complicated. But basically, to do your Punnett square, we take the first parent and we put their genes on the top of the square, 1 letter per box. And we'll say that's the parent that's giving us our egg cells. Half of this parent's eggs will have this gene. Half of this parent's eggs will have this gene. Parent number 2, we're going to put their genes on the side of the square. And let's say that this is where we're getting our pollen from. So half of the pollen cells will contain this gene and half will contain this. Now in this one, it doesn't matter because it's all the same. But if you watch how we just kind of work this like a multiplication table. If the egg is fertilized, this egg is fertilized by this pollen, we get a hybrid tall. If this egg is fertilized by this pollen, we get a hybrid tall, and so on, and so on. And we always, by the way, put the big T first because it makes it easy to do our counting later. So we could ask, what are the chances of getting tall offspring out of this cross? 4 out of 4 of them here will be hybrid tall. This is a certainty. There is a 100% chance we're going to get a tall offspring. What's our chance of getting a short offspring from this cross? Well, 0 out of 4. You have no chance of getting that. When we talk about genetics, it's all about probability. And the only certainties are 100% or 0%. I'm going to show you the next problem where we have a few more variable choices there. So in his next round of experiments, Mendel did a hybrid tall plant crossed with a hybrid tall plant. All right, so we're going to get the eggs from that plant and the pollen from this plant. So once again, we would start with our 4-squared Punnett square. And we put the parent 1, put their genes on the top, again, 1 letter per box. This is parent 2. Parent 2, we put their genes on this side, again, 1 letter per box. And now we have some different situations that could occur here. If this egg is fertilized by this pollen, we would get a purebred tall plant. If this egg is fertilized by this pollen, we would get a hybrid tall plant. This egg is fertilized by this pollen, we'd have a hybrid tall, and these two fertilized, that's where we'd get our purebred short. Couple tips for doing Punnett squares. I always try to make my capital and lowercase letters look very different so that I don't mix them up. All right, so now we can answer some questions about this. What are our chances of getting a purebred tall out of this cross? Our chances there is just 1 out of 4. 1 right there is purebred tall. That's a 1 out of 4 chance. So our chances then would be 25%. 1/4 is 25%, right? What are our chances of getting a hybrid tall from this cross? Well, there we have these two are both hybrid talls. So that's 2 out of 4 or 50%. What are our chances of getting a short? Well, that's right here. Our little t will be our only short one, and our chances of that happening are 25%. So if we add together what are our chances of getting any kind of a tall plant at all, we got 3 out of 4, which is 75%. And so there's where Mendel's 75%, 25% came from. This one will be tall, this one will be tall, this one ought to be tall. So there's a 3 out of 4 chance that you'd have a tall one. And only a 25% chance that you get a short one. So as you can see here, this is explaining these hybrid tall plants are carrying that short gene. And 25% of the time, both parents will give the short gene, and that's when you get the short offspring. Another example I'd like to do for you, just watch one more today if you can hang with me here. By the way, I think I need to give a code word. Oh, I forgot to give a code word at the beginning. Our code word for today, what should it be? Our code word for today is cat. Let's keep it simple. Just watching my cat. I don't know what he's doing over there. All right. So last example of this I want to go over is what happens if we cross a hybrid tall plant with a short plant? What would be our chances of getting a tall or a short pea plant out of this? So once again, we draw the box. All right, here's where I'm going to pause. I want you to try this one. Can you go ahead and put the parents on? This is parent 1. This is parent 2. Can you go ahead and set this up and try this box? Please pause the video and try it right now. All right. That's as big as I can make it. All right. Pause the video and try it, and then restart it after you're done. Oh, by the way, what are our chances of having big T? What are our chances of having big T, little t offspring? What are our chances of having little t offspring? All right. All right, here's your answer. So parent 1, we do 1 letter per box. Parent 2, we do on this side, 1 letter per box. If this egg is fertilized by this pollen, we get a hybrid. If this egg is fertilized by this pollen, we get a purebred recessive. If this egg is fertilized by this pollen, we get another hybrid. This egg is fertilized by this pollen, we get another purebred recessive. So what are our chances of having a purebred tall, a big T? Well, none of these are big T. This is 0 out of 4, 0% chance. It's not going to happen, folks. Big T, little t, we've got 2 out of 4 of those. So 2 out of 4 is 50%. And what are our chances of little t, purebred recessive? Also 2 out of 4 or 50%. All right. So that's the basics of Punnett squares. We can do a lot more genetic problems with these, which we'll be doing over the next few days. Thanks a lot for watching to the end. We really appreciate it and hope you have a